Assalamualaikum and a very good afternoon to Dr. Hafiz and to my fellow friends. My name is Naim and today I will be giving presentations in our class Advanced Food Biochemical with the titles of Emerging Food-Borne Methods of Detections and Biosensor. First of all, this is my tables of content. First is introduction. Second is emerging foodborne, which is foodborne bacteria that can cause illnesses from consumption of contaminated or toxic food. Next is modes of detection, where we have rapid detections, identifications, and monitoring methods of foodborne pathogens or contaminants or toxic. And then the biosensor for the sensitive detections of agriculture contaminant, pathogens, and foodborne toxin. For the introduction, the emergence of foodborne pathogen is a continuing concern for public health agencies and even to consumers. Emerging foodborne are often zoonotics in origin and may include gram-negative, gram-positive parasites and viruses. Emerging foodborne has been defined as the causative agents of an infectious diseases. This is the types of below are the types of pathogens type. There are few emerging foodborne that have been selected, which is Salmonella species, Campylobacter species, Toxin producing E. coli, Listeria Monocytogene, and Norovirus. First of all, we look upon the Salmonella. Salmonella are groups of bacteria that can cause gastrointestinal illness and that called salmonellosis. It is spread by the food handler who do not wash their hands or the surface and tool they use between food preparation steps.
Salmonella sickens 1.4 million people in the U.S. yearly. Salmonella can be found in undercooked meat, poultry, eggs, or unpasteurized dairy products, and foods contaminated during harvest, production, or packaging. Symptoms begin 6 to 72 hours after consumption and include diarrhea, abdominal cramps, fever, nausea, and vomiting. Dehydration is a concern, especially in the elderly and young children. Ask your healthcare provider to take a stool sample. Document what you have eaten in the past 72 hours.
Okay, that is the brief introductions of Salmonella. Next, we move to the Campylobacter Bacteria. It is an infectious agent that can cause campylobacteriosis. It is usually caused by consuming unpasteurized milk, raw or undercooked milk or poultry, or any other contaminated food in contact with feces from infected animal.
Campylobacter is a common bacterial cause of illness in humans. It grows in the intestines of birds and mammals. Symptoms include bloody diarrhea along with nausea, vomiting, abdominal pain, headache, and muscle pain. Symptoms usually dissipate after 1 week, though in severe cases, long-term consequences such as Guillain-Barré syndrome may occur. If you experience any of the symptoms, seek medical care and ask your healthcare provider to take a stool sample. Also, document what you've eaten in the past 72 hours.
Alright, so that is the brief introductions of the Campylobacter. Next, we move to the Ancestral Coli. They are a group of bacteria that normally live in our intestine, and most of the strain does not cause any harms, but some of it produce a toxin that can cause our body to have a diarrhea, such as a Toxin producing E. coli. And this type of bacteria can be found on the raw or undercooked ground meat products, raw milk, and fecal contaminations of vegetable.
E. coli bacteria inhabit the intestinal tract of humans and warm-blooded animals. Most strains of E. coli do not cause disease in humans, but toxic serotypes like O157H7 can cause illness and death. Symptoms such as abdominal pain and severe cramps followed within 24 hours by diarrhea, often bloody, begin 3 to 4 days after ingestion. Prompt medical attention can reduce serious complications. Ask your healthcare provider to take a stool sample. Document what you have eaten in the past 72 hours.
Right, so this is a brief introduction of the Shiga toxin producing E. coli. So next we move to the Listeria Monocytogene. It is a bacterium that causes listeriosis through the consumption of tainted food product, even in refrigerated circumstances. The infection is most likely to sicken the pregnant woman and their newborns, adult age 65 or older, and people with weakened immune system.
The incubation period for listeria ranges from 3 to 70 days. A person may develop fever, muscle aches, nausea, or diarrhea. If infection spreads to the nervous system, symptoms such as headache, stiff neck, confusion, loss of balance, or convulsions can occur. Pregnant women may experience a mild flu-like illness, but infection during pregnancy can lead to miscarriage. A blood or spinal fluid test is required to test for listeriosis. Invasive infections with listeria can be treated with antibiotics.
Antibiotic. Okay, so that is the brief introductions of the Listeria monocytogene. Okay, so next is the norovirus. Norovirus is a very contagious virus that can cause vomiting and diarrhea, and most foodborne outbreaks of the norovirus occur in the restaurants and resulting in contamination that take place during the food preparation.
Norovirus, commonly called the stomach flu, are estimated to cause 23 million cases of acute gastroenteritis in the U.S. each year. Norovirus infects humans through person-to-person transmission or contamination of food or water. Norovirus illness usually develops 24 to 48 hours after ingestion of contaminated food or water. Symptoms include nausea, vomiting, diarrhea, abdominal pain, headache, and fever. Symptoms last 1 to 2 days. However, norovirus can become quite serious in children, the elderly, and immune-compromised individuals.
Right, so that is a brief introduction of norovirus. Next, we move to the modes of detection, where we will monitoring the contaminants in the food product. There are a few types of the monitoring, which is traditional means of assessment. It is a standard method to monitor the quality of food. For example, the inspections of the color, gloss, firmness, shapes, and the size of product, as well as the noting the presence and absence of the visible defect. As you can see from the picture below, we can see clearly the visible defects of the food product. Next is the instrumentation based analysis modes of detections in the food product. There are several instruments that can be used. Some of it is the near infrared spectroscopy for the detections of mycotogenic fungi and associated toxic metabolite. Scanning electron microscopy applied for the inspections of the ultra structural changes. Gas chromatography mass spectrometry, highly sensitive method for the detections of the contaminant, including pesticide and toxin. Next is the biosensor for the sensitive detections of agriculture contaminant, pathogens, and foodborne toxin. A biosensor is a monitoring of qualities of food, and it is considered a key important for many, especially the consumer. For example, herbicides and pesticides are effective on agriculture produce, but extensive use can lead to the chronic toxicity in human hosts during ingestion. Bacterial strains such as Salmonella typhimurian can cause salmonellosis because they are causative agents of these diseases that can lead to the death of ingestion of the product, and consequently the needs to develop the suitable method for these rapid detections of this pathogen, monitoring its distributions of toxin is absolute necessity. As you can see from the Picture below, there's a types of toxin detection of the enterotoxin type A of Clostridium perfringens. Next is the definitions of the biosensor. It is an analytical device that incorporates a biological element for promoting biorecognitions of an analyte of interest. In these cases are the foodborne pathogens and toxin. In the schematics of the biosensor, the first one is the detector, where it is a biorecognition element that have antibody, enzyme, microorganism, DNA, or even cells, where the analyte or sample matrix will bind to them and will produce signal that will be captured by the transducer. There have been few types of transducer: electrochemical, optical, or mechanicals, and then the signal will be output to the readout technologies and amplifier and data acquisition and processing for producing the result. This is the schematic representation showing common working design of biosensors of pathogen detections in food, where we have our sample, whether it's food, humans or animals, or even environment, and the analyte with impurities will be combined with the biorecognition element and will be detected by the detector, and then will given the signal to be captured by the transducer and will be final to the display electronic readout.
A biosensor consists of four major components: a molecular recognition process, a signal generation process, a disposable sensor device, and a reader instrument. Recognition molecules in the biosensor bind to the biomarkers. The recognition molecules are, for example, aptamers, enzymes, or antibodies. The molecular recognition leads to a signal, for example, an optical, electronic, or magnetic signal. All this happens inside a disposable sensor device where the molecules come together. Finally, the reader instrument measures the signal and translates it into information that is meaningful for the user. There are many diseases and many biomarkers, each having different measurement requirements. Also, there are many technological ways how a biosensor can be designed.
Alright, so that is the animations of the biosensors. Next, we'll move to the sensitive detections of the agriculture contaminant, pathogens, and foodborne toxin. There's a few there are few selections of detections that will be having to the biosensor, such as the agricultural contaminant, pathogens, and foodborne toxin. The agricultural contaminants can be contaminants that are released into the environment as a byproduct from the process of the agriculture. Pathogen is a agent that produces a disease, and foodborne toxin is a toxin that produced by the bacteria that can cause food poisoning and also associate with the inadequate food preparation. Next is the detections of the pathogens. For the bacteria, there are several strains that affect the great significance in foods and horticulture industry. That include the E. coli 0157H7 and Listeria Monocytogene as an example. And next is the fungals. The sensitive detections of fungal is a great importance because they are associated with the crop spoilage. For the first one of the detections of the bacteria, the anti-E. coli 0157H7 antibody is labeled with polyaniline (PANI), were immobilized into the nitrocellular strip, as shown in the figures, as shown in the images, and the samples was allowed to migrate up the strip, and these can indicate a decrease in electron transfer from PANI conjugated antibodies. And this is the schematic diagrams of formations of the sensors and its working principles. Firstly, the polyaniline carbon strip and the nitrocellular strip will be immobilized with anti-E. coli 017 antibodies, and when the samples of E. coli was detected in the food samples, it will indicate that by detections of the bacteria. Another example is the detections of the Listeria Monocytogene, which using the rapid surface plasma resonance based immunosensor assay, which incorporate a polyclonal antibody specific for internalin. Internalin is actually a protein on the surface of Listeria monocytogene, and then in this assay format, the antibody was immobilized on the CM5 sensor chips and was varying concentration of cells that were injected over the surface. This is the schematic representations of the geometry and signals transduction mechanism of surface plasma resonance optical biosensors. Next is the detections of the fungal. A key consideration when analyzing fungal cell and spore by optical biosensing are they are significantly larger than bacteria. Hence, it is possible to utilize modified assay formats such as subtraction inhibition assay for the detections of this large fungal spore. Firstly, the pre-incubations of antigen with target-specific antibodies onto the biosensors, and then the unbound of antibody subsequently retained by either centrifugation or by filtrations, and then the enumeration by capturing on the biosensor surface immobilized with antibody-specific capture immunoglobulin, as shown in the images on the left. The observed response is inversely proportional to the amount of pathogen present in the original samples of cultures, and the major benefit of this subtraction inhibition assay is that the system are not exposed to potential harmful pathogens. Next is the detections of toxin. There are a few types of toxin. Firstly is a mycotoxin and water and marine toxin. Mycotoxin are a toxic compound that are naturally produced by certain types of mold, and it can grow on numerous foodstuffs such as cereals. Next is water and marine toxin. The harmful algal bloom that appear in marine and freshwater release different types of toxin that pose a risk to humans and animal health. First, the mycotoxin. Mycotoxin are toxic compounds that synthesize as a secondary metabolite. As example, aflatoxin that produced by Aspergillus species. As you can see from the image showing the aflatoxin molecule produced by the Aspergillus flavus. A brief introduction of aflatoxin. They are highly toxic and carcinogenic as a secondary metabolite produced by the Aspergillus species. They are most potent and carcinogenic that can cause liver cancer. The detections of this mycotoxin by using the surface plasma resonance biosensors, and the principles is based on the detections of the change of refractive index of the medium when an analyte bind to the immobilized partner molecule. This is the same as the bio detections of the bacteria. The biorecognition recognized, selected, analyzed, in this case is a toxin, and then the surface plasma resonance transducer will translate the binding into the output signal, and then the output signal will be read into the readout system, and the analyte molecule in a liquid sample in contact with the SPR sensor bind to the biorecognition element, produce an increase in the refractive index at the surface sensor surface, which is optically measured. Next is the detections of water and marine toxin. Marine toxin pose a considerable threat to human health because of their potential to cause respiratory, neurological, and gastrointestinal problems, including mortality at a very low toxin concentration, which means that this marine toxin is very dangerous even at a very low concentration. For example, the consumptions of the contaminated shellfish meat. this toxin, marine toxin, is produced by phycotoxin, by marine diatom pseudoneisseria pungent that are called as a phycotoxin. There are types of phytoplankton group that can release this type of toxin that can cause poisoning, including a symptom such as amnesic shellfish poisoning. This poisoning is actually a contamination of amino acid molecule called domoic acid. Domoic acid is a neuroexcitatory toxin that produced by this diatom pseudoneisseria pungent. As you can see from the left of the image, pseudoneisseria is a diatom marine that can produce a toxin that will be ingested by another sea microorganism such as shellfish or fish, and then will be consumed by another organism such as human, and then the toxin will be passed into the another consumer. Next is the detections of water and marine toxin. It is reported that a novel electrochemical immunosensor for the detections of the important marine biotoxin domoic acid. The sensor used carbon black modified screen printed electrode CBSPE by using a high throughput method. I also developed a portable 6-channel SPR system for domoic acid detection in clam extract with limit of detections of 3 nanogram, and the portability of this biosensor is a key advantage over the SPR based sensor, because both researchers and governmental shellfish monitoring program have highlighted the need for rapid in situ method for this monitoring biotoxin contamination. That's all from me. Thank you for your attention and thank you for listening. This is my reference. Thank you.